This video covers notes for protein synthesis. Before we can get into the details of protein synthesis, we need to talk about some similarities and differences between DNA and RNA. We have already talked about the structure and function of DNA, or deoxyribonucleic acid, and with protein synthesis, we need to talk some about the structure and function of RNA, which is simply ribonucleic acid. So let's start with DNA because that's what we know. Here are some things unique to DNA. The first is that in DNA, the sugar is called deoxyribose. So maybe let's backtrack a second. Nucleic acids are made up of nucleotides. Nucleotides are made up of a sugar, a phosphate, and a nitrogen base. So for DNA, that sugar is deoxyribose. For RNA, that sugar is called ribose. In DNA, there are 4 nitrogen bases, and B stands for nitrogen bases, and those are A, T, C, and G. For RNA, the nitrogen bases are A, U, C, and G, where U is for uracil. So that is a difference there. The shape of DNA and RNA is also going to be a little bit different. The shape of DNA is, as we know, a double helix. This means it is double-stranded. RNA, on the other hand, is single-stranded. So it's just one strand that can kind of coil up back on itself, but that's another story for another time. The last thing is let's compare the size. When compared to RNA, the size of DNA is big, and the size of RNA compared to DNA is small. Of course, we know they're both small, but RNA is substantially smaller than DNA. So some things that they have in common is that they are both made of nucleotides. And they both have the nitrogen bases A, G, and C. Next, we will look at the function of RNA now that we know a little bit more about its structure. There are 3 different types of RNA that each have a little bit of a different job. So the first one, and the one we'll talk about probably the most, is called mRNA, where the little m stands for messenger. And this type of RNA is made in the nucleus. And we'll talk about exactly how that happens in a few minutes. The second type of RNA is called tRNA, where the T is for transfer. tRNA carries amino acids to the ribosome. And finally, rRNA is ribosomal RNA, and these subunits make a ribosome. All right, so we are going to put it all together when we talk about protein synthesis. So thinking back to the very beginning of the school year, we talked about 4 organic molecules: carbohydrates, lipids, proteins, and nucleic acids. And so nucleic acids kind of hold the information, and we can think of proteins as like executing the information or the things that can actually do something. So the DNA in our cells is, I'm not going to say useless, but it doesn't function on its own to do a task. It is used to make proteins, and proteins can do that task. So maybe the DNA says, to make the enzyme amylase. Okay, and that amylase in our saliva will help to start break down complex carbohydrates in our mouth. So you may want to at some point review nucleic acids and proteins from the beginning to help you kind of get through this. But as always, feel free to ask questions when you have them. So we're moving on to protein synthesis, or how are proteins made. And it happens in two different steps. The first step that happens is called transcription. And the next step is called translation. So we're getting the information from our DNA and transcribing it and translating it into something that can be used. So first we're going to start with the specifics of transcription, because that always has to happen first. Can't have translation without transcription. So during transcription, that is the process of using DNA to make mRNA, or messenger RNA. And this process occurs in the nucleus of eukaryotic cells. That's going to be our focus, but similar type of thing is going to happen with prokaryotes. This process is just happening in the cytosol rather than the nucleus. So a couple of steps here. First, an enzyme called RNA polymerase separates DNA strands. So that enzyme comes along and it's going to open up that double helix. Then that enzyme, RNA polymerase, uses one strand of DNA to make an mRNA strand. Then the DNA will reform, so it's been separated, it's going to be put back together, and then that mRNA is going to leave the nucleus through a nuclear pore. And so here is a little picture of that process happening. You can see our RNA polymerase molecule here is splitting open that double helix, and then it is going to use a strand to make this mRNA strand down here, and then that mRNA strand is eventually going to leave the nucleus. So let's look, here's another picture example of that. So if we know the DNA sequence, we will also know the mRNA sequence. So here's just another picture. This is again transcription. So we have our DNA molecule here. RNA polymerase is going to open up the helix and then add new bases. And it does that by a similar base pairing rule to know what order the mRNA nucleotides need to go in. The key difference is that there is no T or thymine in mRNA. So let's look at our example here. We have a DNA sequence that is T, A, G, T, T, A. That means that our mRNA sequence from that section of DNA would be A, U, C, A, U. And then that information, that mRNA strand, is going to be used in translation to make a polypeptide chain or protein. So here's, maybe I should skip back to why do we even need this to happen. So DNA has all of the information for all of the genes and all of the proteins and all of the things that our cells need to make. But our chromosomes are going to be too big to be able to leave the nucleus. And so we're transcribing that information that's stored in the DNA into something smaller, mRNA, that is small enough to leave the nucleus. So we're transcribing the information from one place to another, and then mRNA can actually be used because it can leave the nucleus. And after it does leave the nucleus, we can move on to translation. Translation is the process of using mRNA to make a polypeptide chain. And that's our a kind of word for protein. And this process occurs on a ribosome. And let's go through our steps on that. So the first thing is that our mRNA is going to arrive at a ribosome and it's going to attach. And the ribosome is going to read the mRNA in groups of 3. Each group of 3 is called a codon. So a group of 3 on the mRNA is a codon. Each codon will code for 1 amino acid. I'm hoping that word is familiar to you. Amino acids are the building blocks of protein. So when we put lots of amino acids together, we can get to a protein. So each group of 3 on the mRNA is a codon. Each codon codes for 1 amino acid. And there are 20 different amino acids that are found in humans. So we do have some repeats. So some, maybe there's 3 or 4 codons that code for the same amino acid. The first codon every single time is always AUG. So that says, okay, time to start translation. I am the start codon, and it codes for the amino acid called methionine. AUG might be found later on in the mRNA sequence, and that's fine, but translation is always going to start at AUG. There will be extra nucleotides on the front end and on the back end of what we actually need to code for the protein for a couple of reasons. One, I guess to make sure that we can actually still get the full protein. So if something happens and we lose a couple of nucleotides on the front end or the back end of an mRNA molecule, we can still get the protein we need. So there's extra nucleotides on the front of AUG and, extra once we get to the end of the coding sequence for the protein as well. Anyway, long aside, then the tRNA molecule with the correct anticodon, which is the opposite of a codon, briefly binds to the mRNA to make sure that the amino acids are put together in the right order. And I'll explain more on that when we get to the picture here. And then the ribosome connects amino acids by peptide bonds. And then translation is going to continue until one of the stop codons is reached. And we'll see what those are later. So there is one start codon, but there are more than one stop codons. It doesn't matter which one you hit. That is when we say, okay, we're done translating. This polypeptide chain is complete. So here is a picture of what that is going to look like. So this gray piece here, this is our ribosome. This is our mRNA strand right here. And then these little tRNAs you can see. Here, let me draw a picture here. So here is our tRNA molecule as an example. This top part has the amino acid like on its head. And the bottom, this is called the anticodon. And so you can see it matches up. Here is our codon and anticodon that are touching, but it briefly matches up so that the ribosome can make sure that all the amino acids are together in the right order. Proteins are seriously picky, and if we do have a wrong amino acid in there, that can make a pretty serious difference. So that is an example, picture example of translation. With our in-class assignment and our other practice this week, we'll figure out, okay, how do we know which amino acid it is, will be there based on what the mRNA strand is. We'll practice with that because I know that chart can be a little tricky. So you do have that to look forward to. Finally, to wrap it all up in one lovely picture, this is, as a whole, protein synthesis. Transcription is going to happen first, where our DNA is going to be separated by RNA polymerase, and then that RNA polymerase is going to make an mRNA molecule. That mRNA molecule is then going to be able to leave the nucleus for step number 2, translation. That mRNA is read 3 at a time by the ribosome, and then that ribosome is going to put together a polypeptide, which will eventually be processed and turned into a protein. Let me know if you have any questions. I know this is a little tricky to, hear on your own without pausing to ask questions. So just make a note of those, and I will be happy to answer them when I see you.